Hello, this is Siuli Mukhopadhyay. Professor Tuhin Shubro has joined.
Yes, I have joined. Uh, Subroto will introduce you here. Professor Subroto Rana will introduce. Then we will start.
Okay, okay. Should I just check once if my slides can be shared now
Yeah,
or
you,
now I can check?
yes, you can.
Okay,
If you
thank you.
wish.
Sorry, ma'am, I was disconnected. And please check your connection and check your presentation whether it is visible or not.
Yes, I'm doing that. Just a moment.
Ma'am, it is visible.
It is. I think yeah, but the whole screen is being shared, so I'm just trying to. Can you see my screen?
Yes, ma'am.
Yeah, but nothing is there in the screen.
Yeah, yeah.
With the permission of Professor Mukhopadhyay and Dr. Tuhin Subra Bhattacharya, should I start now?
Yeah, I don't know whether Professor Mukhopadhyay has completed
Okay,
presentation.
maybe we can wait for 2-3 more minutes. It's just
Yeah,
about 10,
Okay,
10
okay,
here.
okay.
Just let's wait for
2-3 minutes.
two more minutes.
Yeah.
That will be good. Thank you.
There seems to be some problem sharing my screen. Immediately, I'm getting logged out if I share my screen. Uh, are you being able to hear me?
Yes. Yeah.
Yes,
So,
we can hear you.
Professor
Yes,
Suhin, I have WhatsApped you my slides. Is it possible for you to project them?
Okay. Okay. I will try. Yeah. Yeah.
I'm very
I
sorry,
will do
but
that.
I've been typed 3 times and I'm getting logged out here.
So we will take 5 more minutes, and then we'll start.
sure, sure.
I think I can share it now. Can you all see it?
It's visible now.
Yeah, okay, okay. Yes, okay. So it's moving also, isn't it?
Yes, ma'am. Yes, it's perfectly fine.
Okay, okay. Yeah, I'm very sorry. I'm very sorry for the problem. Yeah, okay. So, sh- should I wait
So,
then
so,
for the
should I
introduction?
start now? Good evening, everyone who have joined the session. We welcome all of you to this lecture series for propagation of statistics education and research organized by Calcutta Statistical Association as a part of its outreach program. Calcutta Statistical Association was established in 1945 by a group of faculty members of the Department of Statistics, University of Calcutta. From the very onset, the objective of the association has been to promote activities related to statistics. Using its limited resources, the association continues to play a key role in developing statistics. As an integral part of ongoing outreach initiative, the first session of this online lecture series was organized to celebrate 75 years of CSA's glorious existence during pandemic 2020. And today's lecture is the last lecture of the third session. The fundamental objective of this series is to engage and enrich the knowledge of undergraduate, postgraduate, and doctoral students specializing in statistics and related fields. Eminent statisticians from India and abroad have previously delivered lectures, eliciting a positive response from both the students and researchers. The association has always focused on advancement and dissemination of knowledge of statistics through its various publications. The Calcutta Statistical Association Bulletin, CSA Bulletin, is highly regarded by the international community of statisticians, and since 2016, the CSA Bulletin is being published by Sage Publication. Since 1991, CSA, jointly with the Department of Statistics, University of Calcutta, is organizing an international symposium on probability and statistics every 3 years. And the 11th International Triennial Calcutta Symposium will be organized this year during 27th December to 30th December. It's now time to introduce our speaker for today. Today's lecture will be delivered by Professor Siuli Mukhopadhyay from the Department of Mathematics at IIT Bombay. She is also an associated faculty member of the Quetta Center for Digital Health at IIT Bombay. She is currently leading the National Disease Modeling Consortium. She did her Bachelor in Statistics from St. Xavier's College, Kolkata, Masters in Statistics from University of Calcutta. She completed her PhD in 2006 from University of Florida, USA. She joined Medical College of Georgia, USA as a faculty member in 2006. In 2007, she joined IIT Bombay. Her research interests include theoretical and applied statistics, biostatistics, disease modeling, and health informatics. Today, she will be talking on statistical methods for tracking infectious diseases. There will be a question and answer session at the end of the lecture. One can put their questions in the chat box during the lecture. Over to you, Professor Mukhopadhyay.
Thank you, Professor Rana, for the very nice introduction. And sorry to everyone for taking time to present my screen. I would really I'd like to start by thanking Professor Tuhim Shubro for inviting me to give this talk, and of course, Calcutta Statistical Association for giving me the invitation to give this talk. I am a master's student. I did my master's from Calcutta University long time back, and I learned so many useful things, and I can see so many of my teachers here, some of my classmates here. So I'm feeling very nostalgic. Yeah, so hopefully the new audience, that is our students, our statistics students, will enjoy this talk. And if you have any questions, please feel free to type it up or ask at the end of the talk, or even in the middle. I don't mind. Okay, so hopefully my slides will behave better now. Yeah, thank you. So I'll start it. So today what I'm going to talk about is infectious diseases and tracking of these infectious diseases. So I will start with a very simple slide about what are these infectious diseases, why we need to track them, and then what are the different methods. And of course, since we are all statisticians here, so statistical methods to do this tracking. So this is a joint work of mine with many people over many years, starting from 2015, with Professor Debraj Chakraborty of our IIT Bombay E department, with my postdoctoral students Divya Kappara, Rashmi Tiwari, my PhD student Satish, and many other students and people from my consortium, National Disease Modeling Consortium, and also from the hospital teams, which is the private public hospital in Kolkata and Bombay, KEM Hospital. So as Professor Rana said, at this point I am leading the National Disease Modeling Consortium of India, and that's why I'm so interested in disease modeling, in building our army of disease modelers. So I thought this is the perfect venue to give this talk. Okay, so my screen has changed. Hopefully you can all see it. So let me just tell you something about infectious diseases. Of course, everyone knows what's an infectious disease after the COVID-19 pandemic that we all went through. So infectious diseases are illnesses or disorders, and these are caused by organisms which are quite small in size. So they are bacteria, virus, fungal diseases, or maybe caused by parasites. Now, how does this infectious disease propagate? That is, I mean, if I have an infectious disease, say if I have COVID, how will I transmit it to the other person? Okay, that's what we are interested in, this transmission. If there was no transmission, if there was only one person who had the disease, there would not have been anybody. But during COVID time, we saw that this disease propagated among humans, and then it invaded countries, then it crossed borders, and there was a need to keep track of it. And then many people use modeling to predict, to forecast, okay, when is the next wave going to come, and so on. So there are some diseases which spread through direct contact. This one of the disease we all know is leprosy. Then there are some diseases which spreads through respiratory channel, what you breathe in. Okay, so that's one example, can be influenza. Sometimes we get sick by when we eat contaminated food, like we go to a marriage party and then we eat something and the food is contaminated by some bacteria or something. And one of the most popular disease we know is typhoid. All our mothers used to say, like when we used to have exams, like don't eat puchka because you are going to land up either getting cholera or typhoid. So these are all infectious diseases which we have heard over for many years. Then there are some diseases which need insects or animals for this transmission, for this human to human. So human to human transmission will not be direct. It has to happen through something like a mosquito or some animal or a rat or something. And one of the most popular diseases is malaria. And we all know, all Bengalis know about malaria and dengue and all these things. So many infectious diseases, such as these old diseases like measles, chickenpox, there are vaccines nowadays which can be used to prevent these diseases. So like there's a polio vaccine, we all know about it, and it was very successful in India. So there are vaccines. And then there are some diseases where the vaccines are not there. Like when COVID started, there were no vaccines, and the vaccines had to be made, had to be created in the labs. And in the beginning, what measures everyone took was like washing hands, keeping distance. Then there are some simple measures like using bed nets. Bed nets is nothing but in Bengali we call it the moshari. Okay, so you use it and then you don't get bitten by a mosquito which may be carrying the parasite, malarial parasite, and then you don't have malaria. So some simple measures. Okay. So what do I mean by tracking infectious diseases? So these infectious diseases, they are like, it's like an ongoing process. They are everywhere, all over the world. In some places there's more, in some places there's less, in some places it has become endemic, it is always there, and so on. So an ongoing process. So disease surveillance. So to track these infectious diseases, always a surveillance process is going on, and it is looked over by WHO. And this is an ongoing process that it's going to see if say there is a measles outbreak in Kolkata. So if there's a measles outbreak, then the first 5 or 6 cases, once it comes out in the paper, then they will start a collection of data. But actually this process is always ongoing. The hospitals or some medical centers are continuously collecting the data. They are keeping the data in Excel sheets, and then this information is given to the public health people, and the public health people are taking decisions to reduce morbidity and mortality. Morbidity means sickness, mortality means death. So disease surveillance is just a process. You can think of a process of a data collection process, and a data collection process of what data? Of the disease data. Now once you have this data, once you have got the data, what do you do with it? So what you do with it is you track those infectious diseases. So how can you track? How can we track? So simple is we can just simply count the number of positive cases, count the number of positive malaria cases in a month, in a week, in some place, and so on. Or proportions, we can say, we can try to see how many divided by what's the number of people staying in that region. So proportion of cases. Or instead of just counting, nowadays we are all fascinated by these colorful maps which come out always. So So these visual maps, which like they are good aids, visual aids, the spatial risk maps, they are called the spatial risk maps. There may be other type of risk maps over time, over space, and so on, used by building, taking this disease data which has been collected and drawing these maps. And then the third most important thing is the disease model. So I will be talking more, little bit about the spatial risk maps and more about the disease model. So now, what is this disease modeling? How is it going to help in tracking the disease? How is it going to play the role in the of being a tracker? So on one side, you have the disease surveillance. Just now I talked about, it's an ongoing process of collecting disease data. How many people are falling sick because of this disease, disease A, disease B, disease C, in this place, in that place, and so on, over time, over weeks. So in one place is the integrated disease surveillance, and there's another department. And these people, they want to know, if there are so many diseases, how am I going to control the disease, and how am I going to get rid of the disease cases? How am I going to eliminate the disease? And disease modeling acts as like a bridge between this disease surveillance group and the control and elimination of disease group. And so the disease surveillance group gives the data, and the data is taken and it's modeled, and the control and elimination groups also gives its control and elimination strategies, and these are also fitted in the model and seen, and the disease pattern, the spread, and the growth of the disease is predicted, and the modelers say that this control is positive, this elimination effort is positive. Here, there is a gap in surveillance. And then again, the disease surveillance people start working. If the modeler says that the control and elimination is not so good, it's like only 50%. Like in the COVID, the lockdowns were controls. So when the lockdowns, all the modelers tried to see if the lockdowns were very effective or not. And if they were like say 50% effective, then the public health officers who are in the control and elimination group, they went back and checked if their lockdown strategy is actually good or not. So that's how the disease modeling will act as a bridge between the integrated disease surveillance and the control and elimination of diseases. Now, what are these disease models? Are there only statistical models, or are there any different type of models? So during COVID times, I think everyone noticed there were some models which are called epidemiological models, like the SEIR models, the SIR models, and so on. So these are one set of models. These are quite old models, and then there are statistical models. There is also a interaction of or joint epistat models, and there are data-driven models where you can employ AI ML methods and have lot of data, lot of inputs and everything, and have a data-driven model. So, are different models needed for different diseases? Yes, models need to differ when you go from one disease to another. So, I told you about two diseases, say influenza and malaria. When we are talking about influenza, we saw that the way influenza spreads through the respiratory path, it's different from how malaria spreads. So when we are talking about a model for influenza, it's still quite simple. But when we are talking about a model for malaria, we actually should remember the part played by the mosquitoes other than only the humans. So the models need to be different when we jump from one disease to another. And then there are always non-infectious disease working with the infectious disease. Some of the non-infectious disease we know are the cardiovascular diseases, the diabetes, then cancer, the different cancers. And then will the same model work for infectious, non-infectious diseases? So there are many questions to answer. Do we need different models for different diseases, and so on? So which model to choose? Is epidemiological model good? Is statistical model good? Is the data-driven model good? There are too many choices. So where do we stand? By we, I mean India over here. So India has always been one of the key priority geographies for global disease elimination efforts. So we made a very big breakthrough with the elimination of the eradication of polio in 2011 using the monovalent oral polio vaccine. So also we have made quite a big progress in tuberculosis control, and that was done by engaging the private sector along with the public sector. So I, being the leader of this National Disease Modeling Consortium with all my other faculty investigators, our partner investigators, and all our RAs and students, what we are trying to do is we are looking at 15 different infectious diseases, and we are trying to understand which are the measures to control these diseases, when will these diseases be eliminated from India, and so on. So kind of modeling, using modeling for understanding the elimination status of different diseases. So in the beginning, what I'll do is, so I have divided the whole talk into 3 parts. The first part, in the first part, what I'll do is, I will tell you about only diseases over, we will consider only disease counts over space. In the second part, what we will do is we will only look at disease counts over time, and then we will think and we will see that if we need a model also for space and time, and we will talk a little bit about that. So the first thing which I will show you is the simple spatial risk maps, and this is a work which was done by my postdoctoral student Dr. Divya Kapara, and also we will see a very simple spatial model for measles. So this is a work which has been done in NDMC. It's very simple, it's very textbook model, very textbook spatial risk maps, but still I just want to show you, to give you a glimpse of our work. So before I go to this slide, I will like to go to this slide. So this map, I hope everyone can see this map. So on the right hand side, I have the district level map of West Bengal. So this is the map of West Bengal, and the different colors are, you see, these are the districts which have been colored. Okay, so what are we doing here by looking at this map? So we want to look at measles, the number of measles cases, and there are 20 districts over here of West Bengal, and we have the data for measles for 20 districts from April 2014 to March 2020, just before COVID. So using this data, we want to see, what do we want to see? We want to see if there is any spatial correlation or clustering due of the measles cases due to districts being close to each other, and so on. Okay. So now what do I mean by spatial correlation? So you know already correlation. So I will tell you what is a spatial correlation. So spatial correlation, we are going to take an attribute. The attribute over here is the measles, number of measles cases in all these districts, and we are going to check using some correlation measure and the coloring of the map that if we have two districts which are sharing boundaries or which are very close to each other, then do their measles counts also behave similarly? By similarly, I mean if I have this district and this district, if the measles counts in this one is high, will the measles count in the neighboring districts also be high or not? If that is so, then I will say that there is a similarity or a correlation over space. So that's what I mean by spatial correlation. Simple. So now when I am looking at measles counts, it's not very fair if I just look at the measles counts, because it always will depend how many people are there in these districts. So I need a denominator which is the population. So instead of just looking at the raw counts, what you usually do is you look at the number of positive cases divided always by the population of that place. And then you can use a some multiplier. Over here we use 10 to the power 5, and then you have the kind of a measles rate map. So to understand the map, first let's go through this very simple spatial autocorrelation measure, which is called the Moran size. So the Moran size is kind of like the simplest autocorrelation measure, and you can think of it as just a weighted version of the correlation which you are acquainted with. Okay, so now in this, what are these weights? What are these W's? So these W's are called spatial weights. And these spatial weights, they change by the boundaries. So if this district is I, then the districts which are sharing boundary with it is this. It shares a big boundary, and then this one, and then this one. So if I take this as I, then if these are all the J's, all of them, then I can say that if they so I can take a measure where I say that if they share boundary, let me take Wij to be 1, otherwise 0. So when I am looking at this, then it's a 1. When I'm looking at this, it's a 1. Even if the boundary is small in size, when I'm looking here, it's a 1. But I'm looking here and here, it's a 0. So that's what Wij is giving you. It's the spatial weight indicating how the observations I and J are spatially related, and the way you want it, that you can choose. So you can either use sharing a boundary as your measure, or you can use a distance measure, or you can use the nearest neighbor measure, or anything, whatever you you want to use. And of course, you can use a lot of measures and count I for all of the different W's. So this is what we do. We first find the I, and then we also look at a simple thing which is called a Moran scatter plot. So this Moran size scatter plot is a simple visual tool which allows you to visualize the spatial autocorrelation. So you can divide up the space into 4 quadrants. So you can see that there are 4 quadrants in this spatial scatter plot, and this quadrant 1 is displaying the region which are high. That means that if I is high, which are the J's which are also high. So those points will go into quadrant 1. If you are looking at high low, then which is quadrant 3, then you are looking at if I is high, which are the regions, which are the J's which are low in measles counts. So that's how the quadrants are divided, and this is a very good way to visualize. This has also a big link to the beta parameter in a regression setting, and you can think of Wx as a response variable where the regressor is x, and you can think of this as the beta parameter. Next, we also do a very simple model. It's called a spatial lag model, where the ith measles count or the measles rate is taken, and we take the regressors or the covariates to be the counts which are in the neighboring region, because Wij will be 0 when the regions are not sharing boundary, and when they are sharing boundary, it will be 1. And then you can have covariates in the model, and there is a normal error. So now going back to this picture, what do we see? So we plot the measles rate of a district, and over here this coloring, when it's very darkly colored, that means that the number of measles cases with respect to the population is quite high. So if you look at the graph, then you see that this region is the highest, and the result is that Purbo Mednipur has a population which is quite low compared to the other districts, but it has quite a high number of cases. That is why the rate is coming out to be the highest. So this is a very simple risk map, just a colored map. Then what we do is we perform the global Moran's eye test, and in the global Moran's eye test, so this is the Moran's eye statistic value which we get, and then we perform a test where H0 is there is no spatial autocorrelation, and HA is there is spatial autocorrelation. And when we test it, then we get a p-value which is quite big, and then we say that there seems to be no clustering, and that also matches with the coloring. So if you see the coloring, you see that none of the high regions are surrounded by high, or none of the low regions are surrounded by low. When we have a high or when we have a low, then this eye will start increasing in size. And we do not have such a coloring, and this is what comes out of the spatial autocorrelation test. In the spatial land model also, we get rho, which is the spatial autocorrelation parameter, to be minus 0.04. It's very small, and the p-value exceeds 0.1. So again, we see that there is no significant autocorrelation. So at least in this data, which is the data for 2014 to 2020 over this, so we have just averaged all the cases over this time point, and we are looking at a single map, not over time. We see over there that there are no clusters spatially. And to go further, we also take some covariates, and the covariates are population density, proportion of zero dose children. By zero dose children is that since measles has a vaccine, how many children in those districts did not receive the vaccine. So if there are many children, since it's a childhood disease, if there are many children, then the disease will seem to go up, the number of positive cases, and we see that we don't find much statistical dependence. Oh, we do. We have a zero dose children proportion, which turns out to be significant covariate. However, rho is very small. So this is what our work from NDMC, we looked at a very simple model. Now, what are the questions which can come out from here? This is a very, very simple way we have studied the data. So now questions can come out. How about if we did not have only the measles counts? If we wanted to look at two things in that spatial risk map, how do I do that? So that can be done, but we have not done it. So then there are ways of constructing these maps where more than one attribute can be looked at visually. So then so when we are looking at this map, we are looking at correlation, which is a global correlation. We are looking at all the districts together and trying to see if there are clusters, global clusters. But how about studying only local correlations and corresponding maps? We have not done that. How about trying to find hotspots of diseases, trying to see where if we have a high region surrounded by high, how do I find those? So we have not done that. So we can do that. Then we have data from 2014 to 2020, but the map I showed you was one single map where I took all the data over the districts and just put them together. How about looking at the time component in the spatial risk map? How do I do that? Then I showed you a very simple SLM model for modeling the data. Is there any other way of modeling the data? Those questions should come to our mind. How about the estimation procedures? What type of estimation did I use? Over here we use maximum likelihood, but are there any any other ways to estimate the data? And the most important thing is, in our model, we did not consider time. We just missed out the whole information from 2014 to 20. We just clubbed everything and just looked at only a spatial model. How do I put even time in this model? So these and many other questions can come to your mind. And these are just some of the topics I have taken to motivate you and to interest you in this area. So the next thing which I will show you is time series models, and this I did for malaria. So this is quite some time back I did in 2014-2016 with the KEM team. So the data came from KEM hospital and with my postdoctoral student Dr. Rashmi Tiwari. So this malaria data, it was collected from KEM hospital, and the data we had was patients testing positive for malaria. The duration, as I said, it's quite an old data. So the duration was from 2010 to 2015. So 5 years data, and the data was available to us in weekly format. Now, if you look at the literature where people are modeling malaria, dengue, these are called vector-borne diseases because you need the mosquito to transfer this disease from human to human. These mosquitoes are the vectors. So this vector-borne disease transmission, if you look at the literature, it shows you that these are dependent on the weather. By weather, I mean rainfall, maximum, minimum temperature, relative humidity, and so on. So what we did before we started even doing this work is I called up our meteorology department and I asked, like, in those days you could still call them. Nowadays I don't think you can call them. Nobody has a 22 number anymore. So I called them and I asked them, can you share with me this data? And then they did, by taking a small amount. So then this weather data was collected, and so I had the weather data, and then I had all the positive malaria counts also, and this was over weeks. So I had 308 such observations. So if I just simply look at the data over those 308 weeks, then this is how the data look like. So it seems good. It seems to be good because it seems as if in Bombay, malaria is coming down. There is a downward trend. Every time it seems as if it goes up at a certain time in a year and then again it goes down. So that's called seasonality. So we spot, if I just look at it, then immediately I spot there's a trend and there's a seasonality in the malaria data. Okay. So that's what I write here. So there's a downward trend detected, then there's a seasonality detected. But if I just model the trend and seasonality, is that enough? Or do I need something else? Do I need to consider, when I am looking at the malaria counts of today, do I need to consider the malaria counts of the last week or the week before? So do I have any autoregressive terms in my model? Do I have any moving average terms? And so on. So what's the model? That's what it comes to. So what we did, what I have done over here, I have simply detrended, deseasonalized the data, and I have looked at the residual, and then this is the autocorrelation plot of the residuals. Whatever is left over, that's the residual, and I see there seems to be a correlation. So what to do with that? So then let's start on how we build the model. So now so at this point I did not have much expertise in time series. Like you all, I had just learned time series in some master's level class or in a PhD level class, that was it. So then I realized that there is a problem with this time series I have. All my responses, these are positive counts of malaria. It doesn't really match with the time series data which are there in the usual books. Okay. So if I look at Brockwell and Davis, oh no, that's not matching. That's not what their time series looks like. Okay, so then I understood that there is a problem. So what I saw is that I can't take the normal distribution for modeling this. I have to do something else. So what I understood is that I have to take help of generalized linear models. Now, how do I do that? How do I include trend and seasonality? How do I include the weather covariates? All these questions started coming up. So what we started doing is we started looking up some count time series papers and books, and we found there are many for time series, there are many for count time series, for binary time series, and so on. Then we started looking at that, and then we knew how to handle this data. So to start off with, we knew that there is a downward trend, there is a seasonality. Now, which weather covariates are we going to include? Do we include all of them? Do we include some of them? So we had 4 in our hand: maximum temperature, minimum temperature, rainfall, and relative humidity. And when we did it, then we saw that an initial covariate analysis. So this is not correct. This is not rainfall. This is actually relative humidity. When I wrote it yesterday, then by mistake I wrote rainfall. So relative humidity and maximum temperature came out to be significant. So we knew that these are the covariates which we need to put in the data. And then what we did is, now you have all heard about this training and test data. So what I did is I partitioned the data and I kept most of it for training. So 256 weeks I kept for training and 52 weeks I kept for testing. So what am I going to do with this training and testing? First, I'm going to try out and select a model and fit it to the training data. and then the best model or models. You always go for models. You never go for one model. So I have a set of models, and from there I choose the best one, which has a very good, all the model statistics are good, and then using that or using 2 or 3 good models, what I do is I try to fit them on the test data, and I see now what the test data is giving me. So test data is like kind of forecasting or kind of testing the future data. So that's what I did. So the first model which we took, since it was positive counts of malaria, so we thought of the Poisson distribution. So we started with the Poisson distribution, and the Poisson distribution differed a little bit over here because this is a time series data. So the mean of the Poisson was depending on time. So lambda, we just didn't have lambda, but we had lambda t, and then we modeled this lambda t, and this mean model is the model which we selected. So that was called eta t, and since you have a GLN, so we use the log function. We tried some other links, and then this one turned out to be the best. So the eta t were known functions of the covariates of relative humidity and maximum temperature, trend, seasonality, and then it had also AR and MA terms. So these are the models which we looked at. So in the first part, when we have xt transpose beta, this is just the part where we have the covariates. That is, first comes the intercept, then the time, then the humidity, then the maximum temperature, and this is the part where we have the seasonality. And then if we go back to the model, then these are the autoregressive terms, and these are the MA terms. And so we had all different models, and we tried to see how these models perform. Along with Poisson, we also looked at the negative binomial model. And the negative binomial, the mean and the variance was different. So in Poisson, we have the mean and the variance is same, and I didn't really want that. The dispersion seems a little bit high, so we also wanted to check the negative binomial model. So we looked at all these different types of model under the negative binomial setup, and again the xt transpose beta is the same as before, and we looked at all these different 8 models, and then we looked at all the model statistics, different model statistics, and at the end we thought that the best model which comes out is model 6, because it has very low values of AIC, chi-square and mean square error, and then we fitted the data using the model 6 for just the training part, and we saw it's quite a good fit. Over here there seems to be an outlier. We could have done some outlier analysis. I think we got lazy and didn't do it, but it seems not to be such a bad fit. So the actual counts are represented by the stars, and the fitted values are by the lines. So it seemed to be quite a good fit. And then we looked at the residual analysis, and the ACF and PSF were all perfect. Residual, it passed the test. Randomness and the normal QQ plot also came out to be fine. Next, what we did is, this is for the public health. So how are these KEM people, the hospital people who gave me the malaria data, going to benefit anything from my analysis? So I just can't think of only the statistics part. I have to also think of how my statistics is going to, or my model is also going to help them. So what their need was, tell me forecast, tell me the future, tell me, everyone wants to know the future. So the future they wanted to know is that, when is malaria going to come again? Then if they know that, then they are going to be ready with their hospital beds, they are going to be ready with their number of doctors, with their medicines and so on. In COVID time, everyone was interested in when the next wave is going to come back, because then they are going to be ready with all their resources, and they are going to save people. So that's what they needed from me. So I had to also forecast, and the forecasting I did very simply by one step ahead forecast, and I gave them some forecast, and their forecast was not too bad, and this we have on the test data, and it was quite good. So this is the mean absolute deviation and the standard deviation. So now what are the takeaways from this? So number 1 is that, did I consider the mosquito population? No, I didn't. Okay, so I considered a very simple statistical model. Maybe it was a little bit complicated because it was a count time series and so on. But I did not consider the actual transmission pattern of the disease. So that's still left. Okay. Then what about other covariates? Like, are there any places where there is lot of water or population density? All these covariates we did not consider. All the socio-demographic covariates we could have. Then what about the interventions? In these 5 years, were there any interventions, any control measures by the government? Did the counts go down because of that? In the middle, we saw that the trend is going down. Did the trend is going down just because of time that malaria is going away, or is it due to some intervention measure? We did not model any of that. Then, of course, the question comes, are there any other way to model, any other fancy way to forecast the counts? And what about spatial transmission? We never thought about spatial. We are just thinking about time here, nothing about space here. So we again miss space. Before we had missed time, now we are missing space. So the next thing what I'll discuss very little bit is how about putting both of them together, space and time, and doing something with disease data. So the last thing which I show, very small I'll show, is because we are also, I think, almost over time. So this is a spatio-temporal model for dengue. So this is a dengue data again from Mumbai. The Brihan Mumbai Corporation shared this data with me, and this data was over the different administrative wards of Mumbai. So let's see what are these administrative wards. So this is the map of Mumbai, okay, and all these are the different administrative wards. So there are 24 administrative wards, and over here in this part of Mumbai, it's highly populated. The Dharavi slum is over here. Okay, so so over here, what we looked at again, we looked at weekly dengue cases from January 2010 to 2014. But now we are going to think of both space and time. So that's why we are thinking over wards, and we are also thinking over the time series from 2010 to 2015. So we have a number of weeks over here is 380. So the goal over here was to estimate the spatio-temporal dependencies in dengue count. So the actual goal over here is, are these wards affecting each other somehow with dengue? If the dengue in one place goes up, does the dengue in the neighboring ward also go up, and so on? This is being done over time and over space. So you can think of these 4 nodes as the wards. So we have, say, 4 wards here, and then each of the 4 wards have, so the 4 wards are the 4 spatial regions, and we have time series attached to each one of them, and we want to know the connections over time and over space. So YIT is like the ith time series at time t and the ith ward. So the time goes from 1 to 380, and then we have a multivariate time series now, because we just don't have one spatial region, but now we have 4 of them. So we don't have one time series, but 4 count time series now. So it has become more difficult. So how we model this? So we deviate a little bit from the type of, so we don't deviate a little bit, we deviate a lot from what we used before. So now we are in the parameter driven model world. Before we were in the observation driven model world, and over here we use the help of a latent variable which we denote by x, and that is going to kind of tell me how the dengue rate progresses over time. So again, we use a Poisson distribution, and we have the mean. When the mean, we have in the mean we have the covariates, the weather covariates, again come back, temperature, humidity, and so on. We can have any other covariates, and then there is x, when x was not there before, and x is the latent process which is kind of telling me about the rate of dengue transfer. So we can have a ARP model. So we have, so in the latent process is taking charge of all the time dependencies. So over here we try to introduce a ARP process, and doing that we kind of ended with many parameters, and now we have so many parameters. We have Poisson response, we have it conditioned on a Gaussian variable, and then the estimation process becomes difficult. There are too many parameters, so we have to use something other than the simple maximum likelihood estimation. We have to kind of shrink the number of parameters, and we do all that, but I don't mention that here. So more or less we do a space-time thing. So the questions over here is this again, is this the only way of modeling? The second question again comes up. We never took the again the transmission, the way dengue is transmitted, the actual way in the mosquito population and so on. So how do we bring that epidemiological part of the things in this model? We did not. So how do you do that? So these are all the questions which are left unanswered. But before I end my talk, what I want to show you a little bit about is about our consortium, and I would like to invite all of you to partner with us to engage in collaborations. And if students are interested, they should apply for postdoc positions, for internship positions, and so on. So among our audience, I saw Professor Bhashwati Ganguly is here from University of Calcutta. So she is our esteemed partner. She's working in both RSV and also antimicrobial resistance with us. And we have many people from IIT Bombay, from different institutes, from other IITs, and so on. So you are very welcome to partner with us. We really need statisticians working with us. So a very brief introduction to NDMC. So this is a national effort to model particularly infectious diseases in the context of India. It is at this point of time anchored in IIT Bombay, led by me and Professor Ganesh Ramakrishnan, and it was established in 22 late. And what we are trying to do is trying to get an estimate of disease burden in the population, assess control strategies, disease interventions, trying to understand what are the elimination dates. Our focus is on malaria, on neglected tropical diseases, on tuberculosis, and vaccine preventable diseases. And we know that one of the vaccine preventable disease is measles, COVID, and so on. So we have different type of people from mathematics and statistics, from AI/ML, from fluid dynamics, from geospatial departments, from biology departments, from environmental science working with us, from economics working with us. We have an advisory council, and over here Health Secretary of India is in our advisory council, DG ICMR is there, DBT secretary is there, and what they do is they advise us and they tell us that this is the disease which is of importance, you have to act on it now, you have to model this now, and then we do our modeling work. So these are our team, and these are our research associates, students. We also have a fully functional secretariat, and we need many more students, many more RAs to join us. So please feel free to contact us. We hold workshops. We have already held 8 workshops, and we are going to hold more. And we are collaborating with many divisions in the Ministry of Health, with immunization division, with NCBBDC, which is the National Control for Vector-Borne Diseases, with the Vector Control Center, with State TB Office, with CMC Vellore. We also have some foreign collaborations, particularly with Imperial Emory, Pennsylvania University, and so on, and with the Institute for Disease Modeling in Seattle. And we have just started, so we do have some publication and preprints. We are looking for more, and we are of course looking for all of your help. Thank you.
Any questions from any audience?
can I ask?
Yes, yes, please.
Yes, Shuli. So this is Bhashati. Thank you for a very nice presentation. I'm sure it will encourage many other people to join the NDMC. My question is on the West Bengal data. So that was data on measles, right? So
Yes, yes.
what exactly is the data? Is it people who are coming to some surveillance hospitals and getting tested for measles?
So these are everyone in the community with symptoms and so on.
Yeah, so I guess that was my question, that could there be any testing bias, or are we assuming that everyone has access to the tests?
Everyone has access to the test. Yes.
Hospital-based study.
No, it's not. So it's like a database which they collect. Like if there is one district in say Maharashtra, so all the people who have been tested for measles in hospitals in private and who have tested positive, so those counts over weeks or by weeks have been recorded.
But we are assuming that there is no problem of access, right? We're assuming
Yes,
everyone
yes,
can
of course,
access
of
the
course.
test.
So when we actually model over there, there is a big problem of underreporting of measles, because
Right.
measles is a very old disease and it is not going away, even with over 85% vaccination coverage. So there is a lot of underreporting. So over here we are not taking. This is a very simple model, just which I use in class, and I wanted to show you.
But there are models which could take this into account.
Yes, yes, we have ourselves. We have done okay, under reporting and so on
I
percentage.
see.
Yeah,
I see. Thank you.
yeah. Thank you.
Hello, I am DK Ghosh. Actually, in your model, you have taken training time 252 weeks and test time 56 weeks. Do we always take test time less than the training time?
So when you want to train a model, so you're kind of selecting a model. So you need to keep a bigger percentage of data for the model selection. That's what you do. So what is test? So test is ideally if new data flowed in, then there is no need to do this partition. Then you could have just tested it out on the new data which could have come. So what is testing? I'm going to take the model which I have from my training part, and I'm just going to use it on the test data and see if it fits it well or not. That's all I'm going to do. I'm not going to choose a model there again. So yes, I will say that when we, even in a regression analysis class, I say that 80-20%, because the test data, it never flows in. Whatever we have in hand, we have to divide it up. It should be done randomly, done many times and so on. Over here we simply, it's a time series data, so we take the first few the 252 weeks or whatever you said, I don't remember. So the 252 weeks for the training and the rest for the test. Yes.
But in general, test time we take more than or nearby than training the model in general.
No. So in the test, you are not fitting the model again. You are just using the fitted model to just, you are applying it on the test data and you are seeing if the fit is good or not. You are not again selecting the data. You are not again selecting the model. Sorry. You are not again selecting the model. So when you are selecting the model, you need more data. And when you are just testing it out, you need less data.
Okay, thank you.
Good evening, madam. Actually, I saw that data is showing seasonality. You have applied only auto-regressive. Can we prefer for adding
No.
something?
We have also applied the seasonality, the yearly, the quarterly seasonality. Actually,
Ah,
yeah. So if you look into, wait, just a minute, it's not moving. Wait, yeah. So if you look at that one, if you look at the XT transpose beta, that part, then I had a harmonic term which was, since there are 52 weeks, so it was by 13. So there was a quarterly seasonality. So that was also, seasonality was also there.
okay, madam. Thank you.
You can test out any other seasonality. So over here also we saw seasonality in the first picture. And we did.
I have a question. This is Nitish Mukhopadhyay, Professor Siuli Mukhopadhyay. Nice to see you
Yeah,
virtually.
nice to see you too. Yes.
Yeah, thank you for an excellent talk.
Thank
I
you.
have couple of questions, and I would not say they are really questions. They're more like generic comments. I Could you please show me the slide for where you talked about negative binomial,
Yes, yes, yes,
please?
yes, 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 yes.
Okay. Now, so these are very generic thing, and I'm not particularly asking any question about modeling and, so this you have lambda t. I suppose you were thinking of over dispersion. So you have lambda t, but k does not depend on t.
No, no, no.
Was there a reason for it?
Yes, here for simplicity, but we have also tried to take later in our work, but then it didn't come out to be dependent on T. So this simple K works. It worked fine. So we just left it as it is.
Okay. And was k, the value of k across the board, was it reasonably small or large?
I don't remember. I think it was small. It was not very large. Yeah.
Not very large, small, yeah. But that's what happens in entomology also. K is usually very small, and that
It was
becomes,
not very small, as far as I remember. I can check
I
the
see.
paper. It was not very small. Yeah, I
Okay.
never put the K value here. I'm very sorry, but it was not very small.
Okay.
Yeah, but it was not of much. Like I didn't have to worry much about it, so it was okay.
Okay. But in entomology, they worry a lot about K because K is very uncertain and K changes very fast because K has interpretation. Um, K has interpretation about how things spread across the plane. For example, weed population, how they spread in your lawn, things of that kind. So that's just a concern. And my
Good evening, ma'am. I have a question.
Yes,
Uh,
yes.
Ma'am, thank you for this excellent talk. I really enjoyed it. But I am from a non-statistics background. I am from a geography background and have used a little bit of this spatial modeling during my doctoral studies. But I would like to know certain things, like if I'm having covariates, especially in the spatial context, one where I'm having district level data for entire India, and other covariate if the covariate we are not getting it at a district level, but something like rainfall where it's kind of spread, it is a continuous data all over the map. So how do we incorporate that kind of data into the model? That is my first question. And my second question is, for example, like the map of malaria you showed for Mumbai. So we have the ward-wise data of malaria, and how do we incorporate the impact of maybe any major sewerage line or maybe any canal where maybe there are high chances of mosquito infestations? How do we incorporate those things into the model? Because that also has this connotation of space, and with the increasing distance of space from the sewerage lines, the incidence of malaria, if we can see to it.
Yeah, so the first question is over different spaces. You are saying that one you have district level and one you don't have district level, you have a broader level. So I have not worked in this type of things, but recently in a conference I heard a talk where the time, so space, either space or time. So over there, the time is like some observations are taken over weeks, and then there are some covariates or something which affect, and these are taken over say months. So those type of maybe techniques can be applied. At this point, I will say that what we have done is, so one thing which we did is that uh, we were looking at this measles modeling, and something similar is we found we wanted live birds, and we were not getting the live birds over districts. So we had like live births over states, I think. And what we did is like kind of we found out the populations of the districts we knew, but we didn't know the live birth. So somehow we tried to gauge like what percentage of the total state population is the population of those districts. Using that relation, we put it in the live births. So something like that, if you can take like area of the district, because it's rainfall, so area of that district, if you can find out from that polygon, and
Yes.
the latitude, longitude, and then from there, and the area of the state, and somehow you can try to gauge the rainfall, because the rainfall and that area and how much millimeter of rainfall, I think that's related. So this is like something which just comes to my mind. So this is one. The other thing you are asking is very specific, that is you have some sewerage line. So then I think we have to really go down to the latitude, longitude, whatever we are using. So the sewerage, so if I look at a district, and I look at the center of the district, I have that centroid. Then from there, if I can try to gauge the distance of the sewerage line, and somehow I put that in as a covariate in the model. So something like that you can do.
Thank you. Thank you so much, ma'am.
Yeah, yeah. Thank you. Any other questions?
Hello, Professor Mukhopadhyay, can you hear me? So this
Yes,
is me,
yes,
Dr. Junaid Khan.
yes.
I had one question related to that spatial estimation. Like, you were considering a district level spatial estimation, and district level data sets are available for each district, and then you were trying to calculate the spatial autocorrelation. So now my question is, if you are considering district level estimation for this spatial, checking this spatial phenomena, how far it is generalizable? Like, we are trying to consider, like, calculating the spatial autocorrelation. So if you consider the number of districts for West Bengal in particular, so the, like, it's not very larger number. So how far this autocorrelation measurement is generalizable? That is what I just wanted to know. Like, again, building up a model like in a spatial autocorrelation models, and then adding covariates into that system of equation. So how far this estimation is, like, we can kind of, how far we can go building up this kind of model.
Yeah, so this Moran's eye, which I showed, and the spatial lag model, these are all textbook material. So these are for whatever area you want. The thing is that this is a global measure. So if there are any local clusters, okay, so it may not catch. So there are some problems like that. But if you want to go from 20 districts of West Bengal to 36 districts of Maharashtra, we have applied this only. These are simple. This is not nothing which we made. These are like textbook models, correlation measures, and this can be applied. But what useful information you get from them, from a global measure, or do you need a local measure? Do you need to do hotspot analysis? These are all things which you can keep on doing, more specific according to the area you have in hand and so.
Okay, thank you.
Is there any other question? Thank you, Professor Mukhopadhyay, for your so interesting presentation. I would like to extend our deepest gratitude to everyone who has made this event a success. A special thank you goes to Professor Siuli Mukhopadhyay for her insightful lecture on statistical methods for tracking infectious diseases. Her thorough exploration of spatial methods, including Moran's eye, spatial lag model, use of time series analysis, and identification of disease hotspots, has provided us with valuable knowledge and practical approaches to tackle one of the most pressing challenges in our field. Professor Mukhopadhyay's expertise and clarity in presenting complex concepts have greatly enriched our understanding. We are grateful for the time and effort she has invested to deliver such a talk. As we conclude the third session of our lecture series, we would like to thank all of the speakers of this lecture series, Professor Ashish Kumar Chattopadhyay, Professor of Statistics, University of Calcutta, and former Prof. Academic, University of Calcutta, Professor Monisha Pal, Senior Professor and HOD of St. Xavier's University, Kolkata, and Retired Professor of University of Calcutta, Dr. Surupa Chakraborty, Associate Professor, St. Xavier's College, Kolkata, and Professor Siuli Mukhopadhyay, Professor, IIT Bombay. I would like to express our appreciation to the President, Calcutta Statistical Association, Secretary, Calcutta Statistical Association, and all the live members of Calcutta Statistical Association, and also appreciate to the organizing committee for their hard work in putting together this lecture series. Your dedication ensures that we continue to benefit from the expertise of distinguished professionals in our field. Finally, thank you to all the attendees for your active participation and engagement. Your presence and interest are what makes these events meaningful and successful. We look forward to seeing you at future sessions and continuing our journey of learning and discovery together. In the fourth session, our next speaker is Dr. Soham Sarkar of ISA Delhi. At this time, we declare the session is closed. Thank you and good
Thank you.
night to
Thank
all
you.
of you.
Thank you, Dr. Rana. Thank you very much.
Thank you, ma'am.